Good day, everyone. I am Aliyah T. Amugis, and my groupmates are Cindy Claire Obrero, Cyril Kate Lanoza, Steven Selgas, Roger Jem Kadirao, and Vidal Clark C. Kabahar. Our research topic is entitled Analysis and Synopsis of Moss Species in the localities of Lake Pinamaloy and Lake Apo.
A moss is a flowerless spore-producing plant with the spores produced in small capsules. Moss is a plant in the division Bryophyta. It has approximately 12,000 species that are distributed throughout the world. The moss flora of the Philippines includes about 700 species, about 50 of which, including one endemic genus, Meriliobrium. Although mosses are widespread, many species are poorly documented. Much more research on these critical indicators of forest vitality is needed. Mosses are among the most important plants in the Philippines because of the role they play in forming the soils on high mountains that absorb rain during typhoons. Mosses are typically found in moist environment, although most species can withstand prolonged periods of desiccation. In terrestrial ecosystems, mosses are important in preventing soil erosion.
Hi, I am here to discuss the kinds of moss found in the Philippines. First, we have the Pugunatum macrophyllum dozy and moke. Second, we have the Physidens nobilis grift. Third, we have the Rhododendron gigant swagger. The fourth one, we have the Nechiropsis lepiniana or the mont flesh. The fifth one, we have the Cerhopodon Tristicus nest X squager. And the last one, we have the Hypnodendron vitiens myth in seam.
In this analysis, the goal of the researchers is to understand does studying moss helps the biodiversity of Lake Pinamaloy and Lake Apo?
Hypothesis. Studying mosses will not just help the biodiversity of Lake Pinamaloy and Lake Apo, hence it also provides a habitat to the large diversity of flora and fauna.
The study was conducted to recognize how mosses are beneficial to the biodiversity of Lake Pinamaloy and Lake Apo. Specifically, this study further intends to, first, to assess the different species of moss found in Lake Pinamaloy and Lake Apo. Second, to compare the moss's characteristics between the two lakes. And lastly, to determine if this study will help the biodiversity of the lakes.
Significance of the study. The relevance of the study is to explore the mosses around Lake Pinamaloy and Lake Apo. This study investigates the specific ways on how mosses in different areas differ from each other. It will also focus on how mosses helps the biodiversity of Lake Pinamaloy and Lake Apo. Limitations of the study. This research is limited only to the localities of Lake Pinamaloy and Lake Apo. This study started on February 2022 and it will end on May 2022. This study will determine the different species of moss present in Lake Pinamaloy and Lake Apo and their benefits to the environment.
Methodology. Entry Protocol. This letter of request will be sent to the office of the Punong Barangay of Sur Don Carlos Bukidnon for Lake Pinamaloy and office of the management in Ginuyuran, Valencia, Bukidnon for Lake Apo. Location and duration. The study will be carried out to the vicinity of Lake Pinamaloy, located at Don Carlos Bukidnon, and at Lake Apo, located at Valencia City. These lakes are presented because it is a forest-like surrounding where mosses naturally lives. The target location will be at the most shady and moist parts in the lake. The trees are also included. The researchers will also try to find moss directly into the lakes to find some aquatic moss. The duration of this study is from February to May 2022. Species composition and identification of specimen. The researchers will describe and identify their diagnostic character using a compound microscope. Researchers will also ask experts to help in identification through the confirmation of the pre-identified specimens. Field sampling technique. 2 sampling plots in each lake measuring 20 meters by 20 meters with an interval of 10 meters in between plots will be established using fishing lines. Opportunistic sampling plot will also be included. Mosses inside the plot will be then collected. Collection and preservation of specimens. Moss will be scooped with bare hand or a flat kitchen flipper tool and gloves. When picking up moss, it should be ensured that there are no grass included. Specimens will be pressed as quickly as possible after collection by using a cardstock or a paper envelope. The moss elements will then be stored in a clear plastic container labeled with its locality. Each moss specimen will be numbered and tied with its primary identification, exact location, plant proper, and date of collection. Determination of microhabitat preferences. Ecological data, light, water, topography, habitat, substrate, trees, rocks, where mosses are commonly seen. Statistical analysis. There are 2 analyses used throughout this research, and those are one-way analysis of variance and correlation analysis. Photo documentation. Photographs of the mosses on their original substrate and the study site will be documented by using mobile phones.